Thanks again, Daniel, and thank you to Open Box Science for uh, the opportunity to present my paper and for the interest in the paper. So for viral infections, we often organize CD8 T cell differentiation based on the outcome of the infection. Acute infection leads to T cell memory, chronic infection leads to T cell exhaustion. But early in an immune response, the system has no way of knowing whether the infection will be acute or chronic. This uncertainty raises important conceptual questions like if memory and exhausted T cells arise from distinct developmental pathways, when did these paths first diverge? And also, without a priori knowledge of infection outcome, how is this decision made? Today, what I'll show you is evidence for the notion that memory and exhaustion, right, arise from a shared differentiation event. This event occurs early after acute and chronic infection, and it's designed to prepare the host in advance for a potential chronic infection while also conferring flexibility, the ability to adapt based on how the infection unfolds. So this project started in 2019, and it started just after a series of really wonderful papers had just come out. And what these papers did was they defined in great detail the different T cell states that arise during chronic viral infection. And one of the key points is that there is a subset of T cells referred to as stem-like T cells, and other folks refer to them as precursors or progenitors to exhausted cells. This subset exists, and what's fascinating about it is that it combines properties classically associated with T cell exhaustion, like high levels of inhibitory molecules, with properties classically associated with memory. These cells, even though they're in the middle of an active viral infection and could have been in the middle of it for weeks to months, they retain the capacity to proliferate. They can self-renew, and they can further differentiate. And the most important thing about these cells is that these are the cells responsible for maintaining the T cell response for the duration of the viral infection. So really the project started as a way of trying to better understand how the biology of these cells works. Specifically, the first question we asked was, when are these stem-like T cells first generated during chronic infection? And there's not enough time for me to go through the data addressing this point, but the key point, the take home, was that these stem-like T cells are generated very early, within the first week of the chronic infection. Another point that's made is that as the infection transitions from the early to the late phase, these cells go from proliferating to quiescence, right? And this is an association with changes in a number of molecules related to those processes. Now, I should be clear, our paper is not the first to show that stem-like T cells are generated early during chronic infection. There are a number of other papers that have come out before showing these data, and in particular, a paper from Daniel Utschneider and Axel Calles, as well as another paper from Tochi Wu, spelled this point out in really great detail. But we were curious as to what that observation, though, really tells us, right, about the design of T cell differentiation. What does it tell us that the cell that is required to sustain T cell immunity in the context of a long-term chronic infection, why is it being generated at a time point that's so early that, again, the host cannot know that the infection is possibly going to be chronic or acute? What we reasoned is potentially what this means, or what this represents, is the CD8 T cell response preparing the host in advance, right, for the potential of a chronic infection. Now, what if that were the case? We also reasoned you should find a cell type right, resembling, right, this classic stem-like or T-peck cell, also early on in an infection destined to clear quickly, like an acute infection. So what we did is we used the LCMV infection model, okay, in mice. Okay, we used, of course, as a lot of you will know in the audience, what this model or system allows you to do is to track T cells of the same specificity over time as in the context of two very, very different infections. So on the top row here, what you're looking at is a group of mice infected with the chronic LCMV strain clone 13. Okay, and you can see the dose as well as the route, right? And we compared T cells on day 5 after this infection with T cells on day 5 after an infection, okay, that was destined to clear quickly, LCMV Armstrong. And we used the same dose and the same route. And what's clear, you could see from the flow cytometry here, this is just phenotyping of those antigen-specific cells, is clearly populations of a TCF1 positive, PD1 positive cell that also has tox, and various other phenotypic traits that we commonly associate with stem-like T cells. Now, for those of you who have done this comparison yourself, it's a very common comparison, acute versus chronic LCMV. You'll know that classically, the dose and the route for acute LCMV, or Armstrong, is different than what I just showed you. Classically, it's a lower dose at 200,000 PFU IP. Sorry, and the route then is IP. And what we could see is you, even after this lower dose and route, you could still see a population of these stem-like T cells arising early. We then sorted these stem-like T cells on day 5 after acute and chronic LCMV and compared them transcriptionally and epigenetically. On the left-hand side, you can see the RNA-seq data. And what this shows is that the transcriptional profiles of these acute and chronic stems on day 5 were incredibly similar, differing by less than just 1%. Only 93 genes out of 9,600, okay, were differentially expressed. And on the right-hand side, you see the MA plot for the ataxia data. This is showing that these early acute and chronic stems had 0 differentially accessible regions. So they looked incredibly similar, but did they function similarly? So to address the functional question, we once again did the same setup. We infected 2 groups of mice, one group with that acute LCMV strain, the other with the chronic LCMV strain. Then on day 5, we sorted that stem-like, right, versus the effector cell subsets and transferred them into separate groups of naive mice, right, and then challenged with the chronic infection. And what you'll see first that I'll highlight is that irrespective of whether the stem-like T cell made or came from an acute or chronic infection, in response to that chronic challenge 7 days later, they generated the same number of progeny cells, suggesting, or sorry, indicating, okay, that their expansion potential was the same. In addition to that, when you looked at what types of progeny cells they made, this was also the same. So you could see that if the cells came from acute infection, right, they make the same number of stem-like T cells as the ones that came from chronic infection. And the same thing is true for the effector progeny. Okay, so what this shows so far is that early after acute and chronic LCMV on day 5, you generate, okay, virtually identical populations of stem-like T cells. But to get at that conceptual point I was making earlier, that you generate this population irrespective of whether it's acute and chronic infection, with the point being to prepare in advance for a chronic infection, we would expect that if that were true, then the cells originally derived from this acute infection should be able to maintain the T cell response long term, say if you put it into a chronic infection. So the experiment we did, okay, to test that, so once again, we have a group of mice infected with this acute LCMV strain. And then on day 5, okay, we're sorting out the stem-like versus effector T cells, and then transferring them into separate groups of chronically infected mice, but at the same time, so time matched on day 5. And then we're tracking the fate of those transferred cells at various time points after the chronic infection. And the result, I'll give it to you straight away. This stem-like cell, even though it originated in an acute viral infection, okay, was able to sustain the T cell response long-term across multiple tissues in a chronic infection setting. And it could do this for weeks. Now, in contrast, and as we would expect, the effector cell, if it was derived from an acute infection, was not able to sustain a T cell population. You can see very clearly in multiple tissues over time, the numbers of this cell crash. Now, regarding the progeny of that stem-like T cell when transferred into the chronic setting, what did it do? What did it make? What it did was it maintained a population of stem-like T cells, as you can see here, okay, at multiple time points. And then it also effector differentiated. So again, those two key properties originally defined for these stem-like T cells, the ability to maintain the T cell response long-term, and then self-renewal, right, and effector differentiation. Even though this early stem-like T cell was taken from an acute infection, right, it still did all the things one would associate with the stem-like T cell in a chronic infection. Okay, so to us, that lended support, all right, again, to this idea that you generate a population of stem-like T cells early on in acute and chronic infection, irrespective, okay, of the infection outcome. And if the virus persists, that stem-like cell also persists, transitioning from proliferation to quiescence. I didn't show you that, but in our adoptive transfer experiment, we also note that this transition happens. And that cell will then will sustain the T cell response long term, okay, by generating effector cells as well as maintaining a population of stems. The question that remained was, all right, if you have a stem-like T cell population generated in acute infection as well, what happens, all right, after the virus clears? Does this cell share a lineage relationship at all with classical memory T cell subsets? Or does this cell really just generate it? Its sole purpose is just to maintain the T cell population in the setting of a chronic infection? That was the question. And so we did a very similar experiment, right? But as I just showed you, but the inverse. So we took, all right, a group of mice infected with chronic LCMV, all right, on day 5, and we sorted out the stem-like versus effector cells, and we transferred them into day 5 acutely infected mice, and then we analyzed the cells at 2 time points after the virus had cleared. And what was very clear is that, no, excuse me, these stem-like T cells were not lost when virus was cleared. Instead, they lost their classical properties associated with being in an actual infection and swapped those for properties that we often associate with memory T cells. So let me walk you through the data here. On the top row, we see the pre-transfer phenotype of the cells. So this is at day 5, okay, in a chronically infected mouse. The bottom row, okay, is showing you the phenotype of those cells, okay, at two time points after, okay, the acute viral infection is cleared. All right, so this is a composite of both day 8 and day 15 post-transfer into an acute clearing infection. So the first thing to note, PD-1 expression, right, is lost. This makes sense. Antigen has cleared. What was interesting, though, is the transcription factor tox, okay, often, of course, associated with exhausted T cells and thought to be a crucial driver of T cell exhaustion. this transcription factor was lost whenever the cells were transferred into this clearing viral infection. TCF1, as we expected, was maintained. What was interesting is the IL-7 receptor, an important for the survival of memory T cells, and often used and is used as a marker of memory precursor T cells. Okay, although of course it's not expressed pre-transfer in that chronic infection, what you'll see is that a very clear population of these stem-like T cells upregulate the IL-7 receptor after transfer into the clearing infection. Something else that was interesting was that after transfer into the acute infection, these early stem-like T cells upregulated the L-selectin, right, an important marker for central memory T cells. Now, what's important to note, and I don't want to leave this out, it wasn't just these stem-like T cells, okay, that acquired properties of memory after the virus cleared. Even though we took a TIM3 positive effector cell from a chronic infection, right, and put them right into so even though we took them from that source, when we put them into an acute infection and watched the virus clear, okay, a portion of these cells too also acquire properties of memory. So aligns with an important concept and what decades of work have shown related to effector dedifferentiation as a route to memory. So I'll quickly walk you through this data. So here again is that pre-transfer phenotype of the cells, of the effector cells on day 5 in chronic infection. They're PD-1 positive, they have TOX, they don't have TCF1, they don't have the IL-7 receptor, and they don't have L-selectin. And then you can see after transfer into a clearing viral infection, PD-1 is lost on them, so too is TOX. Very interestingly, a population of these effector cells that I'll remind you again were once TIM3 positive that have upregulated, right, TCF1. They've also upregulated, right, the IL-7 receptor. What's very interesting though is that unlike that stem-like T cell that I showed you, these effector cells, at least at these 2 time points after viral clearance, don't appear to upregulate the L-selectin. So now I'll just quickly summarize in a cartoon what I've just shown. Early after acute and chronic infection, you get this population of stem-like T cells. They start off all proliferating. If the virus persists, that stem-like T cell population is maintained, and it's maintained with its canonical phenotypic traits. It keeps PD-1 expression, it keeps TOX expression. And what it's doing and what the job of the cell is in this context is to maintain that T cell response. Okay, and then eventually it transitions to quiescence as the infection gets to the late phase, something that we think is important for their maintenance and their ability to persist long term. And it gives rise to various effector populations. Now, if the virus is quickly cleared, the cell is not lost. If the virus is quickly cleared, it drops, right, things like PD-1 and TOX, and acquires the IL-7 receptor, in addition to L-selectin. It's suggesting that, right, it's giving rise to states we associate with memory precursor cells and central memory cells. So that's how we came to our title, right? An early precursor CD8 T cell that adapts to acute or chronic viral infection. And that's why we think, okay, that this is an important mechanism, okay, for being able to deal with that uncertainty that I mentioned at the very beginning of the talk. To deal with the uncertainty, you generate this adaptable precursor early during acute and chronic infection, such that if the virus ends up being persistent, you have a way to sustain the T cell response. But if the virus is quickly cleared, right, you then can propagate cells of that same antigen specificity, but in a setting, okay, where virus is no longer around, right, so they can be there if there is a reinfection, right, and they can respond. But they're not the only cells that give rise to memory. Effector cells also do. And then I'll wrap this up quickly now with like a framing or what I think these findings mean for our framing or understanding of how T cell differentiation develops, how memory and exhaustion develop. So early in acute and in chronic infection, there isn't a difference. You generate two subsets, a stem and an effector. All right. In the case that the virus persists, that effector cell is lost. But that stem T cell, as I've been showing you, right, persists and maintains the T cell response by giving rise to these effectors. In the setting where the virus clears, both that's a stem-like T cell and an effector T cell will go on into the memory population. What appears to happen first is they can both acquire a state that we classically associate with memory precursors. By that I mean the expression of the IL-7 receptor without any inhibitory molecules. Another point that I didn't show you in the talk, but it's in the paper, and you can see it very clearly. At least at this time point, your classical terminal effectors or short-lived effector cells seem to be coming only from this effector cell, not from that stem-like T cell. That's what we saw with our adoptive transfer of those subsets into an acute or the clearing viral infection. Another interesting point, those stem-like T cells, again in that clearing infection, what comes from them, in addition to a memory precursor-like state, is also a central memory-like state. Now, then I put various arrows about the relationships between these subsets and then resident and effector memory, which we didn't provide, of course, any data for. These are question marks is the point. All right, and with that, I want to thank Rafi Ahmed for the opportunity to train in the lab. Absolutely incredible.